Good morning, class. Today we are going to study the topic pedigree analysis and genetic disorder. All of the conditions regarding gene axons, either dominant, recessive, codominant, have been obtained from analyzing the result of controlled crosses. But in some situations, we do not have the opportunity to perform control crosses. Rather, we need to analyze an existing population. This is always the case when studying human genetics. Scientist has devised the another approach called pedigree analysis. It is used to study the inheritance of gene in human being. So pedigree analysis is also very useful when studying any population when progeny data from several generations is limited. So pedigree analysis is also useful when studying species with a long generation time. This analysis help in genetic counseling to avoid genetic disorder in the future generation also. So a series of symbols are used. A series of symbols are used to represent different aspect of a pedigree. Once phenotypic data is collected from several generations and pedigree is drawn, careful analysis will allow to determine whether the trait is dominant or recessive. So it is a very helpful process in genetic counseling to avoid the genetic disorder in the coming generation of a family. So now the question is, what are these genetic disorders? A genetic disorder is a health problem actually caused by one or more abnormalities in the genome. It can be caused by a mutation in single gene called monogenic or multiple gene called polygenic. So sometimes it may be monogenic and sometimes it may be polygenic. Sometimes it is found that these genetic disorder is due to by a chromosomal abnormality. There are well over near about 6,000 human genetic disorder that are grouped into two categories. One is Mendelian disorder and another is chromosomal disorder. Now, what are this Mendelian disorder? Mendelian disorders are mainly determined by alteration or mutation in a single gene. It always obeys the principle of Mendelian inheritance during the transmission from one generation to another generation. There are very few examples, very important examples related to the Mendelian disorder like Hemophilia, colorblindness, cystic fibrosis, Sickle cell anemia, Phenylketonuria, Thalasemia, etc. Another disorder named chromosomal disorder. Chromosomal disorder is caused due to the absence or excess or abnormal arrangement of one or more chromosomes. if there is absence of any copy of entire chromosome or excess of any copy, or sometimes there may be some abnormal arrangement of the chromosomes. It is called the chromosomal disorder. Very important, three very important chromosomal disorders are known: Down syndrome, Klinefelter syndrome, and Turner syndrome. In this picture, you are looking some human inheritance and genetic disorder. One picture shows polydactyly. Polydactyly means there is a 6 finger in our hand. It is due to some regions that disorder present in chromosome number 17 sometime. Polydactylity. Another disorder is shown in this picture is hypertrichous, means excessive hair growth in your body on the outer surface of your body. There may be excessive hair growth. Another example is progeria. It is found very rare, but due to the abnormality present in the chromosome number 5. So here we are going to study some very important genetic disorder. Number 1 is the hemophilia. What is this hemophilia? Basically, it is a sex-linked recessive disease, and the defective individual continuously bleed to a simple cut. After getting a very simple cut, there is no blood coagulation. The gene for hemophilia is located on chromosome X chromosome. In this disease, a single protein that involved in the clotting of blood is affected actually. This disease is transmitted from unaffected carrier female to some of the male progeny. In this slide, we find that a chart, a possible outcome for each pregnancy. If a father is hemophilic, H denote for hemophilia, hemophilic, married with a lady which is totally unaffected by the hemophilia. What about the progeny? The chances of progeny is no son will hemophilic because the recessive gene is present on X chromosome of the father. But if the child is female, then all daughters will be the carrier. In the next slide, if a man is totally unaffected, a father is totally unaffected by hemophilia, married with that lady who is the carrier of hemophilia, then what is the possibility of the offspring? Here you see son without hemophilia, 25% chances. Daughter not a carrier, totally healthy, 25%. But the 25% son will be hemophilic and 25% daughter will be the carrier. Another situation in hemophilial disorder. That is, if the father is totally hemophilic, means father with hemophilia married with a mother carrier of the hemophilia. Mother is carrier and father is hemophilic. Then what is the possible outcome? Again, 25% son not affected, 25% daughter with hemophilia, because X chromosome contain the hemophilic genetic disorder. 25% son with hemophilia and 25% daughter with the carrier of hemophilia. So female suffering from the disease only in homozygous condition, and the female become hemophilic is extremely very rare, because mother of such a female at least carrier and the father should be hemophilic. It is only in that condition. Affected transmit the disease only to the son, not to the daughter. Daughter can receive the disease from both mother and father. The next very common genetic disorder is color blindness. It is also a X-linked disorder in which person cannot identify few colors. The normal human retina contains two kind of light sensitivity cell. One is called rod cells that active only in the low light, and another cell is known as the cone, which is active in normal daylight, and both are responsible for color perception. Normally there are three kinds of cells that are the cones, each one sensitive to a specific range of wavelength. Means red cones 64% chances, green cones 32%, and blue cones only 2%. Now what is going to be happen in the color blindness condition? What are the types? Monochromatism 1. The condition of complete color blindness in which all color appear as a shade of grey. All the colors appears as a shade of grey. Second condition, dichromatism, the condition of a partial color blindness. It is known as the partial color blindness in which only two colors are perceptible. Example, red green or blue yellow. Third condition is known as the daltonism. In this condition, the color blind person who unable to identify in between the red and green color. He or she may not be able to identify the differentiation of red color and green color. Another condition of color blindness is protonopia. The color blindness characterized by defective perception of red and confusion of red with green or blue screen. Always found a confusion. Deuteraponia, a form of color blindness characterized by insensitivity to the green color. The person is not able to identify the green color. Look at the picture also. A person is not able to identify the green color. This is the picture by which a person can examine the color blindness condition. One very important and genetic disorder that is known as the sickle cell anemia. Sickle anemia due to the sickle cell. In this disorder, shape of RBC becomes sickle shape actually. The cells die early and caused shortage of RBC. To carry oxygen in human, which cell is responsible? That is RBC. It is a single gene mutation disorder. One particular HBB gene, look at the slide. HBB sequence in normal adult hemoglobin, HbA. It produces an abnormal version of beta globulin that is called the HbS. When HbS is formed, the single gene which is going to form the glutamic acid, it is converted into the valine. Nucleotide CTG, ACT, CCT, GAG. GAG is for glutamic acid formation in the normal condition. But if HBB sequence in mutant adult hemoglobin, HbS condition, GTC is formed, which is responsible for the formation of valine protein. And due to this valine protein, the shape of the hemoglobin cells or RB cells can change into a sickle cell. In this slide, look at the child. He is suffering from Down syndrome. Down syndrome is due to the abnormality in the 21st pair of autosomes. It is also called Mendelian idiocy. In another slide, it is about the Turner syndrome. What is this Turner syndrome? The individual has 2n. 2n is equal to 45 chromosomes instead of 46. Means there is a lacking of a single pair of chromosomes. Such persons are sterile females who have rudimentary ovaries. Means ovaries is there but it is non-functional. Lack of secondary sexual character, short stature, web-necked, and abnormal intelligence. There may be some cardiovascular abnormalities and hearing impairment is found. Such persons which are sterile female. Another syndrome, a very common syndromic problem, is due to the trisomy of one sex chromosome, X chromosome. Trisomy means instead of 2, there may be another copy of X chromosome. It is formed by the union of an abnormal XX egg with a normal Y sperm or normal X egg and obtained abnormal XY sperm. If there is abnormal XY sperm, the individual has 47 chromosomes instead of 46 chromosomes. Means 22 autosomes plus double X and Y chromosome. Such persons are sterile males. They are called the feminized males also, with undeveloped testes, mental retardation, and female-like sparse body, hair, knock knees, long limbs, and with some female characteristics such as feminine pitches, voice, and enlarged breast. Gynecomastia. So these are the abnormalities of human being due to the abnormal chromosomes. Thank you.